Hello, everyone, and welcome back. I know, exams are just around the corner, and that tension is building up, isn't it? But we're in this together, and trust me, we are going to make sure you walk into that exam hall feeling super prepared and confident. So, I've been getting so many of your messages about this chapter, animal cell culture. It seems a bit confusing for a lot of you, but I promise by the end of this quick explainer, you'll think it's one of the easiest topics. So just take a nice deep breath, relax, and let's get started. Okay, let's just break it down to its simplest form. What is animal cell culture? It's exactly what it sounds like. You take some cells from an animal, or even a person, and you grow them in a dish in a lab instead of inside the body. We just have to create a perfect little home for them, with all their favorite food and the perfect temperature, everything they need to be happy and grow. That's it. That's the whole idea right there. Now, you're probably thinking, sir, wait a minute. How is that even possible? How can cells from a living body survive in a dish? That is a brilliant question. And the answer, well, it comes from a truly incredible story. This is the story of a woman named Henrietta Lacks. Back in 1951, doctors took some cells from her without asking her permission, by the way. But these cells, they did something nobody had ever seen before. They didn't die. They just kept growing and dividing and growing, and believe it or not, they are still growing today in labs all over the world. They were called immortal cells, or HeLa cells for short. And these cells, they basically kickstarted this entire field of science, allowing us to study human cells like never before. This was the big spark. So if we want our cells to grow in the lab, we can't just throw them in any old container. No. We have to give them the absolute best treatment. You have to think of it like we're checking them into a 5-star hotel with the best room service and the perfect atmosphere.
First things first, the food. The room service for our cells is this liquid called the culture medium. I like to call it a super thali because it has a little bit of everything the cells need to be healthy. You've got your amino acids, which are like proteins, you've got vitamins, glucose for that burst of energy, and some salts to keep everything inside the cell perfectly balanced. Then there's this special ingredient, serum. This stuff is full of growth factors that basically tell the cells, okay, time to grow and divide. And just to be safe, we add some antibiotics to make sure no germs crash the party.
And what about the hotel room itself? Well, that's a special machine called a CO2 incubator. It creates the perfect environment. The temperature is kept at exactly 37 degrees Celsius. Sound familiar? Yep, just like our own body temperature, so the cells feel right at home. The air inside has a little bit of extra carbon dioxide, which is super important for keeping their food at the right pH level. And it's really humid, so the little cells don't dry out. See? A true 5-star experience. So, how do we know if the cells are happy and the conditions are just right? Well, the super thali medium has a very clever trick. It has a chemical in it called phenol red, which works like a mood ring. When the pH is perfect, around 7.4, the liquid is a happy, bright red color. But if the cells make too much waste and it gets acidic, it turns yellow. And if something else is wrong and it becomes too alkaline, it turns pink. So you can know the mood of the culture just by looking at its color. How cool is that? Cello, let's pause for a quick exam question. This one comes up all the time. Why do we add sodium bicarbonate to the culture media? Think about what we just discussed with the incubator and the pH. The answer is, it's there to maintain the correct pH. The CO2 in the incubator and the sodium bicarbonate in the medium work together like a team to keep the pH stable at that perfect 7.4 level. Make a note of this one, it's very important. Okay, so we've got our cells in a 5-star hotel. They're eating their super thali. They're happy. So what do? They start to divide and grow. They start to build a whole family. Let's see how this family grows. Now, when we first take the cells from a tissue and put them in a dish, we call that a primary culture. It's like the original big family. It has a mix of all the different types of cells that were in that tissue. But then, if we take a few cells from that dish and move them to a new dish to grow, we call that a secondary culture. It's like taking just one branch of the family tree. These cells are much more similar to each other, which is often what scientists need for their experiments. What's really interesting is that these cell families have different lifespans. Most normal cells are what we call finite cell lines. This means they can only divide a certain number of times, say 50 or 60 times, and then they just get old and stop. This process of getting old is called senescence. But sometimes a cell changes, it transforms, and it gets this superpower to divide forever. These become continuous cell lines. And who does that sound like? Exactly, the HeLa cells. They are the most famous example of a continuous, immortal cell line. So what happens when the cells in the dish have divided so much that it gets really, really crowded? I mean, shoulder to shoulder, no more space, and the food is running out. Well, it's time to do something called subculturing. The easiest way to think about it is this. We're moving the growing cell family into a much bigger house with a fresh stock of food in the kitchen. And here's how that moving process works. First, we add an enzyme called trypsin. It acts like a special key that helps the cells unstick from the bottom of the dish so they start floating. Next, we collect all those floating cells and put them in a tube. Then we spin them really fast in a machine called a centrifuge, which makes them all gather at the bottom in a little pellet. We pour out the old food, give them some fresh food, and finally we split them. We take some of the cells and put them into brand new dishes with lots of space to grow all over again. So, through this whole process, growing, feeding, moving, we constantly need to check on our cells. Are they actually alive and healthy? Just because they're in the dish doesn't mean they're happy. So how do we find out? We use a very simple test called the Trypan blue test. Trypan blue is just a blue dye. Now, a healthy living cell has a really strong outer membrane, like a good security guard at the door. It won't let the blue dye come inside. So the live cells stay clear. But a dead or dying cell, its membrane is weak and broken. The security guard is gone. So the blue dye just rushes right in and stains the whole cell blue. So all we have to do is look under the microscope and count the clear ones versus the blue ones. Simple, right? Okay, so we've learned how to grow cells outside the body, how to keep them happy, how to make sure they're alive. But the big question is, why? Why do we do all this? Well, this, my dear children, is the most exciting part. Let's look at the amazing things we can do with this. We can turn these cells into tiny little factories. I'm serious. Using genetic engineering, we can give these cells the instructions to manufacture important medicines for us. For example, a drug called TPA. It's a protein that can dissolve blood clots. It literally saves the lives of people having a heart attack or a stroke. These cells become our little lifesavers, producing lakhs and crores of rupees worth of life-saving medicine. And TPA is just one example. Just look at this table. There's erythropoietin, or EPO, used to treat anemia, factor VIII for people who have hemophilia, and these things called monoclonal antibodies. They are like little guided missiles that we can design to go and attack cancer cells. All of these incredible, life-saving drugs are produced using animal cell cultures. This is the future of medicine, happening right now. And this brings us right back to where we started, with Henrietta Lacks. Her immortal HeLa cells have been one of the greatest gifts to science. They have saved an unbelievable number of lives and helped with so many discoveries. But remember, her cells were taken without her knowledge or her permission. And for many, many years, her family got no recognition at all. This whole story has started a very important conversation about ethics in science, about the rights of patients, and about a very deep question: who owns the parts of our own bodies? It's something really important for all of us to think about as this amazing technology moves forward. So there you have it. Animal cell culture made simple. I really hope this helped you. Keep studying hard. You are all going to do great, and I'll see you in the next one.